Hi guys, this is Tina, and in this video we'll be continuing our chapter on genetics. So this is a very important part of genetics, and it's meiosis, and I'll be walking you through it right now. So first of all, we need to know the basic concepts. So meiosis is the process that halves chromosome numbers and allows a sexual life cycle with the fusion of gametes. Separation of pairs of homologous chromosomes in the first division of meiosis halves the chromosome number. DNA is then replicated before meiosis, so that all chromosomes consist of two sister chromatids. In meiosis, a diploid nucleus divides twice to produce 4 haploid nuclei, which is shown in the diagram next to this. So you first have the parent cell, which has 4 times as many chromosomes as the daughter cell, as the eventual daughter cell, because cells undergo meiosis twice. And meiosis is only occurs in sex chromosomes because, for example, bacteria or other types of organisms undergo binary fission. So stages. There's meiosis 1 and meiosis 2, both of which are shown in the diagram next to this. So in meiosis 1, homologous chromosomes separate, whereas in meiosis 2, sister chromatids separate. And this will be explained shortly. But there is prophase 1, metaphase 1, anaphase 1, and telophase 1, which are part of meiosis 1, and then all of those that are called the same thing, but with the 2, which are part of meiosis 2. So, the 2 very important stages that you will be asked for are prophase 1 and metaphase 1. So, in prophase 1, this is the early stages of meiosis, which involve pairing of homologous chromosomes and crossing over, followed by condensation. So you need to remember this. It's prophase 1 and crossing over, because this is one of the 2 things that contribute to genetic variation. So as you can see in the diagram, the homologous chromosomes are aligned. However, the blue and the red are now crossing over, and this produces recombinant chromosomes, which are seen in the third part of the picture. There are also non-recombinant chromosomes which occur, which does not promote genetic variation. The second process you need to remember is random orientation, and this is in metaphase 1. So this is the second thing that contributes to genetic variation. So orientation of pairs of homologous chromosomes prior to separation is random, which means that there are 4 possible outcomes for each. And as you can see in the diagram, homologous chromosomes can line up in 2 ways during metaphase and meiosis 1. So one from your mom and one for your dad, which gives a total of 4 possible combinations. And this contributes to genetic variation. So, genetic variation. The halving of the chromosome number allows a sexual life cycle with fusion of gametes. So now we have the crossing over, if you remember, and random orientation, which both promote genetic variation. And this is all caused through the fusion of gametes. So, yes, now you can just see a little summary in the diagram. There are multiple reasons why offspring are genetically different from their parents and one another. And that's why you see that you don't look exactly like your mom or exactly like your dad. And siblings, unless they're identical twins, don't usually look exactly the same. So, once again, to recap, alleles come from 2 parents. So they each donate their own genetic material. There's crossing over, which produces a mixture of maternal and paternal genetic material on each chromatid. And finally, the random assortment, where the homologs and sister chromatids distributed to each daughter cell during mitosis are a random mix of maternal and paternal genetic material. Thank you for listening to this video. We hope it helped.